As promised, we're leaving the endomembrane system. We're going to enter into some different organelles. Specifically, we're going to look at the mitochondria, the chloroplasts, and the peroxisome. Just because we're leaving the endomembrane system does not mean that these organelles are not membrane bound. Remember your definition of endomembrane system. Remember what its jobs were, what its functions were. Remember the very definition of how they were interrelated. These organelles that we're coming up on still have membranes. They're just not part of that system. Okay? So first up, well, actually, let's grab some definitions first. So mitochondria and chloroplasts are responsible for energy. So specifically taking energy from one form into a different form, a more usable form for that organism. So mitochondria are the sites for cellular respiration. Cellular respiration is a metabolic process that is responsible for generating ATP. We will find mitochondria in both plant and animal cells. Chloroplasts are found in plants and algae, okay, so not in animal cells, although I'm still thinking that'd be mighty cool. Not found in animal cells, plants and algae only. These guys are the site of photosynthesis, okay? So we're going to make something here. We're going to make glucose, or a form of sugar here. Peroxisomes are oxidative organelles. Peroxisomes are very similar to lysosomes in that they break stuff down, but they do it by a different means. Okay, so I think of lysosomes as garbage collectors. I think of peroxisomes as very specific recyclers. Okay, so mitochondria and chloroplasts. Remember, they are not part of the endomembrane system, but they still have a membrane. In fact, they both have double membranes. They have proteins inside of them that are made by free ribosomes. So we're going to see proteins inside that are made by free ribosomes, and they each contain their own DNA. Remember when we spoke about nuclei, I said that it was most of the DNA within a cell and certainly the chromosomal DNA, but mitochondria and chloroplasts also have DNA within them, and not the DNA that is active in the process of mitosis or meiosis. So mitochondria are responsible for energy conversion, chemical energy conversion. So we find mitochondria in nearly all eukaryotic cells. Some of them will have more, some of them will have fewer. They have a very smooth outer membrane, and then they have an inner membrane as well. Remember that it's a double membrane, so they have a smooth outer and an inner membrane. That inner membrane is folded into what we call cristae. Singular is crista, plural is cristae. The inner membrane then creates two compartments. It creates the space between the outer and the inner membrane called the intermembrane space, and then on the very center, right at the core inside the inner membrane, we have the mitochondrial matrix. You're going to see that in this image right here, so let's just do that quick. So I have this smooth outer membrane, and then I have this inner membrane, and the inner membrane has folds. Those folds are called crista, or plural is cristae. Okay. Inside here, I have the mitochondrial matrix, and within that mitochondrial matrix, I'm going to have some free ribosomes, and those free ribosomes are going to be capable of protein synthesis just as any other free ribosome is. I also have this intermembrane space. I think I've lost my... Here we go. I have an intermembrane space in between the outer and the inner membrane. Okay, so let me go back. Some of the metabolic steps of cellular respiration, specifically the Krebs cycle, also called TCA, happen here, and then we also see oxidative phosphorylation or electron transport chain with chemiosmosis. Those are going to happen within the mitochondria. Okay. Why do we have cristae? Why do we have those folds? We need a lot of surface area, and anytime I take something and I fold it back and forth, I'm going to increase the amount of surface area. I need that surface area to be able to use ATP synthase as part of oxidative phosphorylation to generate ATP. So that's why it looks like that. Or that's why we believe it looks like that. Okay. Chloroplasts I'm just going to give you a quick peek and then we'll come back to the words as we talk about it. So here are the chloroplasts, and you've got this image in your text. So chloroplasts, we're going to talk about some thycaloids. We're going to talk about a stack of thycaloids being a granum. We've got an inner and outer membrane. We've got some jelly-like stuff in there called stroma, and then again with the ribosomes. So backing up, chloroplasts belong to a family of organelles called plastids. Okay. Chloroplasts specifically have green pigment. That green pigment is called chlorophyll. They have enzymes and other molecules, and what all of that together does, that chlorophyll and the enzymes and the other molecules, their job is photosynthesis. Okay, so we're going to find chloroplasts in leaves and other green organs of plants and algae. So pretty much anywhere it's green, you've got chloroplasts. Okay. They have some structures. So we have stacks. I like to think of them like Oreos. So I have these Oreos stacked on top of one another. Each individual Oreo is a granum, and then a whole stack of Oreos. Each individual Oreo is a thylakoid, and then each stack of Oreos is called a granum. Okay. And then there's some liquid around them called stroma. I heard another instructor use a pancake analogy for you. I use pancakes on Golgi, but this instructor liked to use them here. So a single pancake was a thylakoid, a stack of pancakes was a granum, and the stroma was the maple syrup all surrounding it. Whichever one works for you. Okay, so remember, this is a chloroplast. It's the site of photosynthesis. Okay, our last one in this group are going to be peroxisomes. And peroxisomes are going to do something called oxidation. Oxidation is a process whereby we use oxygen to break down different types of molecules. So when we talked about hydrolysis and polymers, we used water to break up large units, polymers, into smaller units, right? So in oxidation, we're going to take something large and break it up into smaller units, but we're going to use oxygen to do it instead. So what peroxisomes are this single membrane bound little packet that has some specialized enzymes in it. What they do is they produce hydrogen peroxide. Hydrogen peroxide is not good for the cell, but then it undergoes a conversion process to turn it into water, which is fine for the cell. So peroxisomes have a very specific process by which they work. That process is oxidation, and they use oxygen to break up larger molecules into smaller ones. So at this point, we're going to pause again. You should be able to compare and contrast, remember similarities and differences between mitochondria and chloroplasts. You should also have a general idea of what function the peroxisomes play and how they accomplish that.